Good morning to all of you. Myself Nidhi Verma and I am from 11th C class. Today I am explaining my assignment which is made on the topic of locomotion and movement. So this is my first and introduction page. This page is actually a folder which I covered with the help of black chart paper, and on the pink color sheet I have written the details of my introduction. By using the strips of silver glitter sheets and by making flowers, I decorated my whole page. All the pages of this assignment is decorated by dots around the sheets by using the punching machines. So this is my certificate page, and you can see this circle. This circle is drawn with the help of paper cutting and by cotton. I draw this snowman and shade this picture, so you will be see that these types of drawings in every pages of this assignment. So this is my acknowledgement page. In this page I am thanking my principal ma'am, Ms. Sushma Suhag ma'am, and my biology teacher Mrs. Urvashi to give this opportunity to express my deep sense of gratitude to all those peoples without whom this project could have never been completed. So this is my content page, and in this page I written the topics of my assignments. Now I would like to give the introductions of locomotion and movement. Basically locomotion and movement are related to each other, are related to motion but different from each other. For example, in paramecium, cilia helps in the movement of food through cytophanes, and in locomotion as well. These two terms, locomotion and movement, which is linked by stating that all locomotions are movements but all movements are not locomotions. Now we are going to discuss amoeboid movement. It is affected by streaming protoplasm, that is pseudopodia. As you can see in amoeba, this is pseudopodia which helps in the capture of prey as well as in locomotion. Now we are going to study muscles. Muscles are mesodermal in origin, and based on their location, muscles are of three types: skeletal muscles, visceral muscles, and cardiac muscles. First we are going to study skeletal muscles. They are voluntary in nature and they have restricted appearance, hence called striated muscles. Visceral muscles, these muscles are present in the inner walls of hollow viscous organs. They have not restricted appearance, hence called unstriated muscles, and also they are involuntary in nature. Now the last muscle is cardiac muscles. These muscles is present in our heart and are involuntary in nature. Now each skeletal muscles is made up of number of fascicles, and these fascicles are held together by connective tissue layer, that is fascia. These muscles fibers are lined by plasma membrane called sarcoplasm, and in the sarcoplasma matrix is present, that is called sarcoplasm, which contain many nuclei. So this is epimysium, which is the layer of the muscles, outermost layer of the muscles. Perimysium is the outermost layer of muscle fiber, that is fascicles. These are fascicles. Muscle bundles, also known as muscle bundles, these are gel lines. These are muscle fibers called muscle cells also. They are sarcolemma, blood capillaries, and sarcoplasm. This picture is the diagrammatic cross-sectional view of muscles. So this is anatomy of muscle fiber, and this is a sarcomere. So a study of microfibrils has established that the striated appearance is due to distribution pattern of two important proteins, that is actin and myosin. Actin, you can see this I band. I band is light band, and the dark band is myosin, that is myosin protein, and it is also known as A band. Now actin filaments, what is the difference between the actin filaments and myofilaments? As you know that actin filaments are thin as compared to myosin filaments. So in the center of each I band you can see this line. This line is known as Z line, which bisect the I band, and in between the A band the presence of line which is called the M line. In a fasting stage, the edges of thin filaments, that is isotropic, on either sides of thick filaments, that is myosin, partially overlap the free ends of thick filaments. Now we are going to study the structure of contractile protein. So this is, each actin filament is made up of two filamentous actin helically bound to each other. This is F actin, this is tropomyosin, this is troponin, which is also, this is a myosin monomer. As you see that this is head part, this is cross section, and this is tail. So the tail region is called the light myosin monomer, and this is heavy myosin monomer, head plus cross arm. So head is also actin binding sites and ATP binding sites. So each myosin is also a polarized protein. Now we are going to study mechanism of muscle contraction. Now, during skeletal muscle contraction, the thick filament slides over the thin filaments by repeated binding and releases myosin across the filament. This whole process occurs in sequential manner. Muscle contraction is initiated by signals that travel along the axon and reach the neuromuscular junctions on motor and plate. The neuromuscular junction is a junction between a neuron and the sarcolemma of the muscle fiber. As a result, acetylcholine is released into the synaptic cleft by generating an action potential in the sarcolemma. The generation of this actin potential releases calcium ions from the sarcoplasmic reticulum in the sarcoplasm. The increased calcium ions in the sarcoplasm leading to the activation of actin sites. Calcium ions bind to the troponin on actin filament and remove the tropomyosin wrapped around actin filaments. Hence, actin filaments, active actin sites are exposed, and these allows myosin head to attach to this site. In this stage, the myosin head attaches to the exposed site of actin and forms cross bridges by utilizing energy from ATP hydrolysis. The actin filaments are pulled. As a result, the axon reduces. It is at this stage that the contraction of muscles occurs. After muscles contraction, the myosin head pulls the actin filament and releases ADP along with inorganic phosphate. ATP molecules bind and detach myosin and cross bridges are broken. This process of formation and breaking down of cross bridges continues until there is a drop in the stimulus, which causes an increase in calcium. As a result, the concentration of calcium ions decreases, thereby masking the actin filaments and lead to muscle contraction. Thank you to listening me. Thank you and have a nice day.